Yeah, now it's fine. Okay. A very good evening, one and all. It gives me immense pleasure for me to be over here and to moderate this section. I would like to thank Tamil Nadu Science Forum for giving me this opportunity. I have been associated with this forum for quite some time, but not very closely. But now it has been. It's okay.
Hello. Check.
Hello. Okay. So first of all, I would like to introduce the topic. Today's topic is a very, very interesting topic, not only for biology people, but for the general audience per se. So today we are going to see about genome sequencing of extinct human beings. And this is basically due to the Nobel Prize which has been awarded for medicine this year. So let me just give you a brief idea about the topic and the Nobel laureate who won the Nobel Prize for this particular topic. So this year Nobel Prize for Physiology has been awarded to Svante Pääbo for his discoveries concerning the genomes of extinct homins and human evolution. Through his groundbreaking research, Svante Pääbo established an entirely new scientific discipline called paleogenomics. Through his pioneering research, Svante Pääbo accomplished something seemingly impossible, sequencing the genome of Neanderthal man, an extinct relative of the present-day humans. He also made the sensational discovery of the previously unknown homin, Denisova. Importantly, Pääbo also found that gene transfer had occurred from these new extinct humans to Homo sapiens, following the migration out of Africa around 70,000 years ago. This ancient flow of genes to present-day humans has physiological relevance today, for example, affecting how our immune system reacts and so many other processes. Just to give an idea about Svante Pääbo, he was born in 1955 in Stockholm, Sweden. He completed his doctoral studies at Uppsala University in 1986, served as a postdoctoral associate at the University of Zurich and University of California. He established the Max Planck Institute for Evolutionary Anthropology in 1999, where he continues to be involved till date. So with this brief introduction to the topic as well as the Nobel laureate, now I will request upon Professor Krishnaswamy to introduce the distinguished speaker. Thank you.
Hello, can you hear me? Yeah, actually, the earlier that introduction could not be heard at all. The voice is very low. I think when speaking, they have to hold the mic closer and speak or something, because nothing could be heard. So, shall I go ahead with the introduction of Dr. Thangaraj? Okay. So, welcome everyone. It's a pleasure to be able to welcome Dr. Thangaraj who's currently the director of CDFD. Very graciously, he agreed immediately to give this talk following the award of the Nobel Prize announcement. And Dr. Thangaraj did his BSc from Madras University, then went on for his MSc and PhD from Madras University. And he has been working in CCMB for many years and is currently on leave as a director of CDFD. He is a very apt person for giving this talk because he has published many papers, high quality papers in Nature and other journals regarding this, looking at how genome sequences can be used to trace the distribution of humans, and he has been responsible for this, looking at how in Andaman Nicobar Islands people had come there and what is the tracing their spread, and also in the major classification of the two groups of the South Indian and the other group, and then showing that there has been a admixture of them for many thousand years, and how that has led to stratification. He has subsequently been also doing work in this area, and in India, I think he is one of the most apt person to talk on this particular thing of phylogenomics, or looking at how this genome sequences can help understand the spread of humans in the world. So, Dr. Thangaraj, thank you again for accepting immediately, and we hope to listen to your wonderful talk. Thank you, Dr. Thangaraj. Yeah.
Thank you very much, Professor Krishnasamy. It was my pleasure to speak in this forum, and also thank you for inviting me to speak here. What I'm going to talk for the next maybe 50 minutes to probably 1 hour is that how the ancient DNA, which was started and successfully analyzed the genome, particularly the hominin, for the very first time, and how it has been carried for the last maybe 2 decades, which has given a missing link between human and probably the non-human primates, which won the Nobel Prize. And also going to touch upon towards the end that how in India, particularly my own group, has been working on this field, and how some of the conclusions are very useful, not only for the scientific community and also to a higher level between countries and so on. And this field, everybody is very curious to know that how our own species have evolved, how it got well spread across the world. But the main theme here is the evolution, understanding the evolution of human. And if you talk about evolution or human origin, and you all know about Charles Darwin and his work, and he wrote a book in the year 1858 called The Origin of Species, where he has described about almost all the organisms, right from orchids to whales. But what was missing in that book was how human must have originated. Another evolutionary biologist, Thomas Andre Huxley, he wrote a book called Evidence as to Man's Place in Nature in the year 1863. I think that's the very first book which touched upon the human evolution, where he has shown probably this evidence that human must have evolved, human and ape, from a common ancestor. Later on, anatomical, the Neanderthal was also found, which I'll come to that a bit late. With existing sequencing information and various anthropological information, it's very clear that human and chimpanzee have evolved from a common ancestor called hominids about 6 to 7 million years ago. Still, many people are not really believe on this, that how human might have evolved from chimpanzee. And even some of the people who are at the responsible place, they talk that they have not seen chimpanzee or monkey turning into a human. But it's not the correct statement, because the evolution takes millions of years. It's not a magic that chimpanzee or ape or monkey within a minute or a few hours will become human. It takes about millions of years, and been shown very well that this divergent period took about 7 million years. But still, if people are not convinced about evolution, and of course, the genetic changes is the one which bring the evolution. So whatever genetic change takes place in our genome, and it reflects on our phenotype. So genetics and evolution goes together. And if even somebody doesn't like or doesn't understand that there's no evolution, we did not come from chimpanzee, but you look at these three brothers, basically they were from Mylapore in Chennai, but I don't know where are they. I know about 20-25 years back when they were in Bangalore in one of the circus company. I went there to collect sample, but that time the owner of the circus company refused to give the blood. But if that was collected at that time, now it would have been used for sequencing the whole genome of these three brothers, which would have unrevealed some of the mysteries where the DNA changes, which probably brought our features like the way we look like the modern human, and probably there's a back mutation which brought back this kind of features. There are a few more examples. You look at this person and the face and the body full of hair, and his own daughter again, you can see the hair on the face and everywhere. So this is one of the features which we have lost during the course of evolution. Probably there's a back mutation which brought back the hairs and also the feature like chimpanzee and face wise. So definitely there's a genetic basis for bringing all the changes on our phenotype. There are some genes and the mutation, those genes have been very well demonstrated that those are the mutations which probably helped in evolving the modern human, or the particular features which are responsible for what we are having today is because of the mutation in some of the genes. Like the FOXP2, that is essential for human speech and language, that has a set of mutations which is not there in chimpanzee. The mutation in myosin heavy chain gene, which probably created more space in the brain, therefore skull, therefore the brain has grown because of the space in the skull. And there are many more genes which will not go into detail because of limitation of the time. So I'm coming back. What I mentioned diverged from a common ancestor called hominids, which took about 7 million years. But in between, there are several species have appeared and disappeared. The timeline also is given here. And to make it much better, closer, I put it in 2 panels. And you can see that this is a present time and 7 million years ago. So this species was probably for about 1 million years, 6 to 7 million years. So in this process, what are the main features we have gained? We have gained how to walk with the 2 legs. It's called as bipedalism. Then we gain the ability to make tools for survival, maybe killing an animal or hunting for food and so on. The third important feature is increase of the brain size, which I mentioned earlier. There are several genes which have been identified, which are responsible for that. And if you look at that particular character of the brain size, in the early species, chimpanzees had the brain size are almost same as chimpanzee. In Australopithecus, the brain size was predicted that 35% of chimpanzee. In Homo habilis, 50%, Homo ergaster, 60 to 70%. And the close species, Neanderthals, which had a slightly bigger brain than human. But nobody knows whether they are more intelligent or not. But this species is no more. But in one of the paper, Professor Paul Mellers from Cambridge, he has written that one of the reasons for vanishing of the species could be the inherent violent behavior of modern human. Probably true, what we see around. If you look at where these species were, Neanderthals, and they were in most part of the Europe, wherever there are the dots, and the name of the place was given here. So this is the Neander Valley where it was found for the first time. And also some part of Middle East. They extinct, and the age estimate, I can tell you a bit later, maybe around 500,000 years back. And the work has started, or the first identification of Neanderthal skull and skeletal was around 150, 160,000 years back. The first Neanderthal remains was found in Belgium around 1830. Then in Gibraltar in 1848. The Neander Valley specimen, which was found in 1856, was the one actually used for sequencing, and at the time only it has been named as Neanderthals. And this is the cast of the cranium of Neanderthal, and this is a reconstructed picture, and you see that the features, particularly the front side features, are different compared to the human. There are some prediction about Neanderthals because Neanderthals were found throughout most part of Europe, and it was predicted that probably they are the direct ancestors of modern Europeans. Probably they contributed some genes to modern human, or they were completely replaced by modern human without contributing any genes. We'll come to these 3 points a bit later. Now we look at what is the big deal about analyzing the ancient samples. The fresh sample, if you get in a world, thousands of year old, it would undergo the cells would undergo various process, and will have DNA cross-link, hydrolytic lesion, and so on. Ultimately, the DNA will become fragmented. There's base substitution, a lot of contaminations with the bacterial DNA, a contamination with the modern DNA. That's even more dangerous, that when you try to analyze the ancient DNA, all the modern human DNA would amplify, and that would take over rather than amplifying the ancient samples. So I'm not going to detail out the technical aspect of that, but what I'll give you is to say that how the whole process is done. The DNA is isolated from bones, tooth, hair root, and so on. Particularly the petrous bone, the jaw bone, is a very good source for the DNA. And using this drill, make the bone into powder, then process it. There are many processes, not that every sample one can use the same procedure, because the procedure will change based on the nature of the sample, decalcification and many more process. And either if you wanted to amplify a particular small fragment, then you design primer, amplify that, or you amplify the whole DNA for next generation sequencing and capture and sequence and analyze. This is a process which I told very simply, but this is the most difficult part. Once you have the DNA, then analyzing and comparing with any other genome is much, much easier. So this is a very, very difficult part. It will have the endogenous DNA, that's the DNA of the organism, then some contamination outside DNA, and so on. Bacterial DNA. Then when you do the process, the DNA generally what we get is highly fragmented DNA. And there are adapters which can ligate there, then use primer specific to the adapter, capture and sequence it. The very first ancient DNA was isolated and successfully amplified and compared with the modern human was done by Samantha Pabo in the year 1997. He amplified the mitochondrial DNA of the bone, and the mitochondrial DNA is circular and it's 16.5 kb. He amplified only the control region. That's where we see more variations. It consists of two hypervariable regions because it's a more variation, that's why it's called as hypervariable region. Hypervariable region 1 and 2, and 1 will have much more variations. And this is the actual sequence, this is modern mitochondrial DNA. There are two panels. And when you assemble both, wherever there's a dot, that indicates there is a difference between these two sequences. For example, there's a G here, whereas in the other sequence has A. This sequence has A here, whereas other sequence has G. Same way here, there's A and G. So these are the variations between individuals. The mitochondrial DNA is used for many reasons. One is it has a high copy number. If you take one single cell, you will have only 2 copies of the nuclear genome. One copy being inherited from mother in the form of egg will have applied genome. Same way, the other half come from the father in the form of sperm. So they fertilize, then become 46 chromosome or 23 pairs. Whereas the mitochondria, if you take in a single cell, there will be large number of mitochondria. Again, in each mitochondria, there will be a large number of mitochondrial DNA. So that means 2 copies of nuclear genome versus hundreds of copies of mitochondrial genome. So you have plenty of such genome. Then it is circular, doesn't undergo frequent degradation. Then it is maternally inherited. If you want to trace maternal lineage, we have to use the mitochondrial DNA. These are the reason we used to use the mitochondrial DNA. Of course, now we have gone up to the whole genome sequencing for the same purpose. So this is what he has used, particularly he has used sample from the right humors of Neanderthal, and he sequenced, and this is the alignment, this is a reference sequence, and these are different Neanderthal specimens, and wherever there's a dot, that means it's the same sequence of the reference sequence. Wherever there's a different nucleotide, that means there's a C in the reference sequence, T in the Neanderthals. Same way here, there's a A in the reference sequence, G in the Neanderthals. These are the variations which found in Neanderthal when compared to the human. One can also use the nucleotide change to calculate the age of the divergence. So based on that, he has come to the conclusion that Neanderthal and modern human have diverged around 550,000 to 690,000. So that means approximately 600,000 years back, or 500,000 years back. So he was in Munich at the time, then he moved to Leipzig, and where he created Max Planck Institute of Evolutionary Anthropology. And there he has initiated the Neanderthal Genome Project. There are several participants, particularly those who are working on ancient DNA, not many groups. And within Max Planck Institute, there are few, including Mark Stromking and few more. And then he has sequenced the nuclear genome of Neanderthals in the year 2006 with the help of Roche 454. That was the next generation sequencing equipment which was available those days, where you could get only the partial sequence of the Neanderthals. So that sequence, that sample was about 38,000 years old, and fortunately that was free from the contamination of the modern human, because that was probably found somewhere in the cave, and there was no modern DNA contamination. Therefore, it doesn't have to bother too much. And again, it came to the conclusion that the average divergent period is about 500,000 years. Parallelly, at the same time, there was another group. Of course, Pabe was also part of the group. And again, the divergent period, if you look at, say, 706,000 years to 370,000 years. Okay, so on average, if we take, it will be around 500,000. So two independent groups have come to a conclusion by sequencing the partial Neanderthal sequence, or I don't know how much this group has sequenced. Around 500,000 years back, it has diverged from a common ancestor. And there was a write-up, News and Views, by David Lambert. He was in Auckland. Now he moved to Brisbane in Australia. And he explained about both the methods. One is using the virus sequencing and assembling that onto the human sequence. The second one is making amplification, making library, using human DNA as a probe and capturing all the Neanderthal fragment, DNA fragment from the library and sequenced. Then in the year 2010, that was the first time that the whole genome sequence, draft sequence of the Neanderthal was done by Pabo's group, along with David Reich, one of my close friends and collaborators from Harvard Medical School. And they have obtained Neanderthal specimen from 4 different places. And they sequenced, and they also sequenced 5 individuals' genome from different places. Of course, San and Yoruba from Africa, Papua New Guinea, that's close to Australia, Han Chinese, and the French. And try to compare that how much of each of these individuals carrying Neanderthal genome. And found that the Yoruban and San from Africa doesn't carry any of those genome, whereas these 3 individuals were carrying. Subsequently, again, Pabo and his group, they found a new species which is close to human, that they have named as Denisovan, that was found in Siberia. In the cave, there is a small finger. They sequenced and they found they're all human, these are all Neanderthals and Denisovan. This divergence is approximately 500,000 years back, and this is about 1 million years back. So that means human Neanderthal divergence much before there's a Denisovan divergence. Of course, not much work has been done on Denisovan and also not been found in many. Here again, they sequenced the whole genome of, they came to the same conclusion. And this is a place, the cave, it has been found that all the three species, Neanderthal, Denisovan and human, probably must have lived in the same place. Subsequently, the year 2016, David Reach led a study where I am part of it, Samantha Pepper also part of this, and we have sequenced 300 whole genome. We contributed sequence from Indian side, and when we compare the human genome throughout the world, and we found that what is the contribution of both Neanderthals and Denisovan to the modern human. So we found all out of Africa human carrying the Neanderthal genome up to 3%. Right? So Africans 0, and you can see this is a scale, and towards East Asia you have more proportion of Neanderthal genome compared to other places. And similarly, the Denisovan, they have more towards East Asia, PNG Islands, up to only 0.5%. So that tells that there was an admixture between human, Neanderthal, human and Denisovan. Right. Then coming to what else Pabo did apart from finding the link between human and Neanderthal and human and Denisovan. During the COVID period, he looked at the people who are very severely affected with COVID and people who are normal. He took a sample from UK hospitals where we found large number of South Asian, particularly Bangladesh, are with very high frequency compared to the local Europeans. Then he looked at about 50 megabase regions which we inherited from Neanderthals, and he found that the South Asians carry that 50 megabase region with very high frequency, about 40 plus percentage, compared to the Europeans only 16%. Then he came to the conclusion that the South Asians are more prone for COVID infection because we inherited the Neanderthal 50 megabase region. But we all know that when compared to any other population, Indian population is very unique. And we also know that India was doing much better than any other country in terms of COVID infection. Then we try to look at how it is in Indian population. Then we try to see there's no difference. And of course, then we have written a paper and show that the major risk factor which Samantha Pebo suggested that there is a severe for COVID infection. We didn't see that in this. So that gives some kind of suggestion that so nobody can talk about India because it's not like other countries that without knowing in depth of the population structure. Then interestingly, again Pavo found that the chromosomal segment in chromosome number 12, it felt that that gives a resistance against the infection. He found that about 22% reduction when the people carrying those segment which we inherited from Neanderthal, which is spread all over the place. So again, it's contradicting from his own prediction regarding COVID infection. In the recent past, so one of the reasons why probably was been awarded Nobel Prize is not because of only the ancient DNA. The ancient DNA work initiated, demonstrated, establishing the technology and connecting the missing link and continuing that work. So in the recently, I think September, they published a paper where they found that there's a one amino acid substitution in TKTL1 gene, which actually responsible for the human neocortex development, which is very high, producing more neurons in the human compared to the Neanderthal, okay, in the frontal lobe. So that's one. And again, few days back, 19th October, the same group found about the social aspect of Neanderthals, felt that they lived like the human with the social structure. So all of this has won him the Nobel Prize for Physiology or Medicine in this year. Okay, so what now I am going to take you through is, let's first look at some of the views about Neanderthals. It was thought that Neanderthals are the direct ancestor of modern human in Europe. So one of our study, again led by David from Harvard Medical School, where we are part of it, analyzed 8,000-year-old bone from Motala, 7,000 from Stuttgart, and 8,000 again from Luxembourg. So we used both the modern human and the ancient sample to understand the origin of Europeans. Again, we were part of it, myself and Pavo, in this big study, where the conclusion is that there are three founding populations for Europeans. One is the indigenous hunter-gatherer. The second one is Middle Eastern farmers who might have migrated to Europe about 7,500 years back, who might have taken the agriculture to Europe. The third one is North Eurasia, which are genetically connecting both Europeans and Native Americans. So this tells that there's no contribution of Neanderthal absolutely they are not the founder for the present day Europeans. Of course, there are many more things which I'll not talk about because of the time. Then now I wanted to also highlight that how the Indian science towards this line, particularly our own group, is addressing one of the important aspects of modern human origin and migration. So we all know that modern human originated in Africa, and there were two hypotheses that how did they migrate out of Africa. So one of the hypotheses that the modern human must have taken the northern land route, but others don't believe that probably the climatic condition in Europe might not have favoring that migration, therefore they might have taken the southern coastal route. These are all based on the archaeological evidence present in many parts of the world. Unfortunately, we don't have any good archaeological specimens. Of course, we have some which I'll tell you in the coming slides, but there's no strong archaeological evidence to suggest that when did the modern human arrived in the Indian subcontinent. But we have been looking at various populations, and one of the population we all know, some of you might not be knowing that there's a population looks like typically like Africans in Indian subcontinent. They are none other than the Andaman tribes. We have analyzed them, collecting blood sample from the existing Andaman tribe. This is the Unge tribe. And we also looked at the bone sample of Andamanis. Of course, this bone sample not in India. It is in Dept Museum in UK, Cambridge. We collected some sample from there. This happened during the British time. So when they wanted to construct a cellular jail, they killed many of them and took the skull. And we don't know what was the reason that the time, and taking the skull to their museum. And of course, that has helped us in analyzing the bone samples. And we came to the conclusion by analyzing large number of samples that the Andaman tribes are probably the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 to 70,000 years back. So this is the very first evidence using the genetic data by looking at the contemporary population suggesting the early human migration. Of course, in the same issue of science, there was another study by McCullough and his colleague, and they analyzed the population called Orangasi. And they also come to the same conclusion that the early human probably migrated through southern coastal route. And there was a write-up by Peter Foster mentioning our own study and McCullough's study that this is probably the best possible route for early human migration. And I wanted to give you some view about the ancient DNA studies from India, particularly from our own laboratory. This I want to highlight. There's a place called Rupkund in Himalaya, which is about 5,000 feet above the sea level. Most of the time, the water is frozen, and there are hundreds of bone samples. About 60 years back, Anthropological Survey of India has tried to get some samples from there, and they collected and kept in their own laboratory in Calcutta, not been analyzed. And the question was that there are several stories. Who are those people? And there are several hypotheses or kind of speculations. And one of the speculation is that they're ancient traders who are going towards Tibet, and there are some natural disasters happen, everybody perished in the lake. The second one is Zorawar Singh's army from Jammu who tried to capture or invade Tibet in the year 1841. Again, something happened, they all perished. Another one is the king of Kanuj, his wife and all his attendants, they went there, something like a holiday trip, again, some natural calamity happened, and they all perished then. The next one is victims of epidemic, but that's very unlikely, because somebody dies and why they have to take 5,000 feet and to dump it to the well or in the lake. Another one is victims of ritualistic suicide, but again, we are not very sure. So these are some of the speculations, and we have analyzed a team from Hyderabad, from CCMB, and some of the anthropologists and archaeologists also went to collect samples, and we have analyzed, initially looked at how old those bone samples are. They are not very old. There are two sets of samples. One set is around 800-year-old. Another one is 1,800-year-old. And you can see that this is a place where the lake is there. Then we sequenced mitochondrial DNA, Y chromosome, and some part of the nuclear genome. And we came to the conclusion there are three set of samples among the some 37 samples we have analyzed. One set is of the same local. Okay, these are Indians, and root bone samples are matching here. The second one is mostly the Greek and Europeans. Third, there is only one sample close to Southeast Asia. And based on that, we came to the conclusion, because leaving the local people, and they are the Mediterranean migrants, we don't know why they have gone there. A lot of, again, speculations, which we need to address by analyzing more number of samples. Another successful ancient DNA work was from Rakhigarhi. You all know that the modern civilization took place in Harappan region, and it's full of archaeological sites, and our collaborator Dr. Shinde and his colleague, they have excavated and found one intact skeleton. And luckily we could isolate DNA. It's, of course, not very old, 3000 BCE. And we compared with the existing sequence, not only from India, also from outside. And we found that this is Indus Valley sample or Rakhigarhi sample. This consists of Andamanese DNA, and also consists of Iranian hunter-gatherers, but not the Iranian farmers. This is one of the view that the agriculture probably must have come from Iran, and there is no contribution from Iranian farmers, whereas Iranian hunter-gatherers. The sample is only 2000 or 3000 BCE, but this component of more than 10,000 BC. So only one sample, if you get more sample, that would give much more information. Then this is another big study where again we are part of that. More than 500 human remains from both South and Central Asia. And we compared, it is not giving much clue about their contribution to early human migration, but what it says is that they are the steppe postulates, and they probably taken the Indo-European language to Europe, and also brought Indo-European language to northern part of India. The small dark blue component, which again is not very old, 1000 to 2000 BC only. Okay. So not much information was drawn from there. Then this is a very recent one. There's a place called Ajnala in Punjab. In one well, there are a large number of bone samples. Predicted that they are from our army, Indian Army. This happened during the well. We analyzed the genome, mitochondria, Y chromosome, and also small part of nuclear genome. And we found there are specific haplogroup, mitochondrial haplogroup. If you compare that, these are West Eurasian, these are South Asians, and East Eurasians. We compare the different geographical region. one from Punjab itself, Gangetic Plain, Central India and South India, and also with Pakistan. And this signature is matching almost with the Gangetic Plain, but not with the other part of the country, suggesting that although that happened in Punjab, but they probably, and there's some historical evidence saying that people have been recruited from the Gangetic Plain and they were working in Indian army. Finally, just I wanted to tell you about how the Indian, our analysis would have led to something to between two countries, and at political level it had helped. So listen to this background which is there in this video, so that I don't have to tell, it will be more better to listen this. After that I'll continue my talk.
Queen Hyderabad, great-granddaughter of King Constantine of Kartli, ruled over Georgia in Eastern Europe. In 1630, the Emperor of Persia, Shah Abbas I of the Safavid dynasty, conquered her lands and threatened to destroy Georgia. Queen, to protect her subjects from annihilation, surrendered and was taken prisoner in 1640. She remained a prisoner of the Shah until 1624, during which attempts were made to convert the Queen to Islam and force her to join his harem. Both bribes as well as torture were used to persuade her to relinquish her Christian faith. Queen Hyderabad, however, withstood all the attempts and was finally killed on September 22nd, 1624. Her death is also mysterious, as one account says she was crucified to a tree, while another says she was strangled. Legend has it that a light appeared on her body, and the Patriarch of Georgia declared her a martyr and the story, however, doesn't end there, and it is Bessus to Goa in 1627. It is these relics, her right arm and hand, that have come under attention, as it is believed that they were buried in the chapter chapel, the Augustinian convent, and may still be there.
The search for St. Ketevan's relics started in 1989. However, efforts in earnest began in 1998 when the Catholic Patriarch of Georgia searched for the relics of the Queen in Goa. Subsequently, a Georgian delegation, which included a bishop, came to Goa but failed to locate the remains.
Selma Rego relied on the manuscripts of Augustinian friars written in 1870, 1723, and 1629, and in them, in a scientific manner, described in detail the tombstones in the church floor, beside the chapels and the chapter chapel.
Using archaeologically sound techniques, the ASI have cleared the ruins, and in their labors have recently chanced upon the most likely resting place of these relics. This is the second window on the epistle side of the altar of the chapter chapel, above which, according to the book of Silverego, were once entombed the remains of Queen Ketevan. Entombed in the black box, along with Queen Ketevan's remains, were the relics of Fray Guilherme Santa Agustin and Father Jerónimo del Cruz. The ASI came across remains of the black box and a longish bone fallen below the second window. And this bone be the right arm of the saint.
Yeah, so that was the question, because the Georgian government wanted to take back the bone, and the Archaeological Survey of India working hard to trace the bone. And finally they traced the bone. This is the bone, And part of that has been brought to CCMB, and we worked on that, of course, with utmost care to sequence the mitochondrial DNA, and we were successful in sequencing and try to compare with 22,000 of mitochondrial DNA sequence which is existing in our own database, and we found these are the places where the samples have been collected, and none of the samples are matching with the signatures which we found in the bone sample. Then that's one clue that the sample not belongs to India. The second one, the sample is a female, because in the STR marker, these are the various STR markers, and there are X and Y specific amelogenin markers also existing there. So in this, you can see there is only X specific amelogenin is amplifying, but not the Y specific, confirming that it's a female. Then we asked Archaeological Survey of India to get some samples from Georgia. So they got 30 samples, and of 30, 2 samples are matching with the sequence of the bone. So the 3 evidence, one is this, 2 out of 30 samples are matching with the signatures of the bone. The sample is a female, and we didn't find any of the individuals in India carrying that signature. So based on that, we came to the conclusion the relic found in Goan church, probably of the Georgian queen, Ketevan. Ketevan is her name. And this was in 2014. So I don't know how it took so much time to hand over the bone to Georgian government. In last year, last July, our External Affairs Minister, Mr. Jaishankar, has handed over to his counterpart of Georgia small relic of the bone which found in Goan church, and this is one of the extreme example. One can say that the utility of the technique, right, how it can go not only to the scientific and also at the level of international and level of between government and political. So this is the last slide, and with that I thank again for the opportunity to speak. I hope that you all had some clue about the whole aspects, and I'll be happy to take if there is any question. Thank you very much.
Thank you, Dr. Thangaraj. It was a nice talk. Yeah.
Hello.
Somebody has any question?
Yeah, we have some questions. So let me start with my question. So you said this branching out of the Neanderthals and the Sapiens happened some 5 lakh years back. So what happened between the 5 lakh years to the current? We still continue to be the human beings, or it's something in between the Sapiens or the ancestors?
Yeah, last 500,000 years, there's nothing was found. Until we don't find, then we can say that there's nothing, because we never know when the new discovery comes, right? And we all know that the modern human, the way we look like today, so that happened about 2 million years ago, or 200,000 years ago. So we don't know. Before that, there's a kind of 300,000 years. So anything would have happened, but we don't have any clue to tell what happened.
That means we can call them as sapiens, or still they are someone before sapient stage?
Oh, they are Neanderthal. They have been named as a Neanderthal, Homo Neanderthal.
Okay.
Any questions? So first of all, thank you very much for the lucid talk. And I have a very basic question.
Can you please come closer to the mic?
Yeah, you are able to hear me?
Yeah, now better.
Yeah. So thank you very much for the lucid talk, sir. Like you covered almost every important aspect of the Neanderthal genomics. I have a very basic doubt. So recently there have been some enthusiasm about finding out whether all humans actually evolved from Africa and migrated to other parts of the world, or this evolution of human actually took place at several different continents parallelly, especially in the southern part of India, like where people are speculating like there is a submerged continent like Lemuria, where human evolution could have also taken place. So what would be your take on that one, sir? The Indians, have they migrated, or was they were indigenous population who evolved here?
Yeah, the multicentric origin people have proposed, but there is no evidence for that. So finally, it's Africa is the place where everybody have rooted. So Indians, of course, I didn't tell much about our Indian population because of limitation of time. We, in another study, what we have shown that the early human migration, that is the Andamanis, where they were traveling through coastal routes, some stayed in southern part of India, whom we call as ancestral South Indian. They are the founder for Indian population, particularly in South India. Similarly, there was a population for North India, we call them as ancestral North Indians, and they were kind of admixture between these two groups between 1900 years to 4000 years. So there's no evidence so far to support the multi-centric origin of modern human. We all migrated from Africa.
That's nice to hear. Thank you, sir.
I want to know whether this genetic study is only related to human species only.
Please come closer to the system, because that mic is not audible to me.
I want to know whether this genetic study is only related to human species only. What about the other species like birds, other animals and snakes and like that? And do we apply, I don't know the methodology. Are you going to apply the same method to find out the origin of different species? So, or do you stay only with that human? Because it's a very complicated subject, evolution of not only the humans but other species. Why do anybody is interested in those species also? Because there will be interrelationship between studies of different species. There is interrelationship. I think we can learn a lot also in those mutual understanding of genetic study of different species. I don't know. Do you think about it?
No, not to think. It's already being done. Because I was talking only about my work and about the Fabos work which won the Nobel Prize. There are institutes exclusively to do animal genomics, exclusively to do plant genomics, even within India. So a lot of work is going on using the same technique. This is whatever I have been talking. So it is being already done. Maybe next time you can invite somebody from plant or somebody from animal, bacteria and so on. So it's a parallel is going on. The technique is the same. How we make use of that is the one which is important.
One person from Zoom. Madhavan, can you please unmute and talk or ask your question?
Hi, Thangaraj. Good talk. Nice hearing you again.
Thank you.
Just wanted one clarification. I know there are a couple of labs in Tamil Nadu also doing the ancient genome sequencing, like Dr. Kumarasamy. And so, do you have any idea? No, he always tells me that DNA is more contaminated with more bacterial genome and so on and so forth. So, are you anyway helping them or just guiding them for make a clean ancient genome sequencing from Kiladi or any other excavation project in Tamil Nadu?
Yeah, we have been in the discussion with Kumarasamy and particularly the state archaeological department. And in fact, they also invited us to visit. That was the COVID peak time where we could not go. So I think it will be ultimately done in collaboration with us. I don't know how much it was done already. And in the recent past, we have not discussed about that, but we have been discussing about how to analyze the Kiradi sample particularly. Then we also started a little bit of Achanalu sample, which was collected by Archaeological Survey of India and kept in their museum, which is in the Tamil Nadu State Assembly campus. I went personally and collected some samples from there.
Okay,
Thank you, Thangaraj. Thank you.
Some questions from the Zoom's chat window. So, who was the first single gene species after which amphibians start to live outer of water? And would be any kind of certification for this? Yeah, certification will be given. The question is, who was the first single gene species after which amphibians start to live outer of water?
That he has gone back to several thousands of millions of years back. So there are several species because the genus itself different. The one I vaguely remember, which I studied in zoology, that after fish, then the amphibians, then the reptiles, they slowly came out of water. But the question is not very clear.
Thank you. So I had two questions. One is, I believe, actually, contrary to previous discussions, a small amount of Neanderthal DNA has been found in Africans, much less than people out of Africa, but a small amount has been found. So that was my first question. Is that true or not, and how do you explain it? The second question is, what happened to the Neanderthal Y chromosome?
Yeah, Y specifically is not been studied, but it could be the same as, almost same as the human. But of course, we don't know the position of the genome, because if you take the Y chromosome of human and chimpanzee and other non-human primate, there are a lot of reorganization. Some of the genes which are present in the short arm, like SRY in human, that is found in the Q arm of some of the non-human primates. So there could be a lot of rearrangement, but in the overall, in the autosomes, they have aligned perfectly, and also because the human and Neanderthal interbred, I forgot to include that slide. So there they found, unless otherwise the chromosome numbers are equal, then producing the next generation is not possible. So they also shown alignment of chromosome-wise sequence of both human and Neanderthal. So Y chromosome exclusively they didn't say anything, but I think it's almost like a human.
Any question? Yeah, please come here and ask. So before it come, I go on now. Yeah.
Thank you, sir. Okay, my question is regarding the Neanderthal genome project in 2010, actually regarding that. And many gene pool studies shows that the ancestors have mating with the two different groups, like told that Neanderthal and actually the Denisovans. So in your opinion, it may be right or not, that Denisovan and Neanderthal may be mattered or not, I think.
Yeah, they also met. That's what the one paper it was shown. Somehow that slide is missing. So this is small, tiny proportion of Neanderthal genome was found in Denisovan. So Denisovan genome also found in Neanderthal, and I showed that both Neanderthal and Denisovan also found in human. Because probably the cave where all the three species live together, so that probably is explained.
So, thank you, Dr. Thangaraj. I am Vijayan. I was coordinating with you on this lecture. So I have one more question with regard to your work, which is not directly connected with the Nobel Prize. This is something related to the theory of out of India population that was ruled out. What was the evidence that there was a strong feeling that Indians are migrated outside rather than someone come to the India. Now, how was it ruled out?
No, out of India migration is not ruled out, right? The origin of people from India probably ruled out. Right?
Correct. Still, we are migrating. That's happening.
Yes.
But
so if you look at the Australian Aborigines, and they are matching with Andamanis, because we have shown the route, right? People migrate from Africa through India, Andamanis, and there's a population called, in Kerala, Kurumba. So Kurumba, Australian Aboriginals and Andamanis, they all share some kind of genetic affinities. Okay, that's one towards this side. Towards north, if you see the European Romas have actually migrated from India about 1,400 years back. So, of course, in the recent past, there are several migrations. So.
One more question from Zoom. Was there an intelligence explosion after the introduction of Neanderthal genes in humans?
I think fully the question, so you say that...
Let me read the question again. The question is, was there an intelligence explosion after the introduction was there an intelligence explosion after the introduction of the Neanderthal gene in humans? No, he said no. So there is a mixture of genes, in Neanderthal genes mixed in the human genes. Because of that, is there any intelligent explosion?
So you cannot say intelligent explosion, but you can say that the intelligence made the people to survive, right? So in those days, unless otherwise they are intelligent and physically well fit, they would not have survived, right? Those are some of the factors which are naturally selected the individual to survive.
May I read the next question from the next question is the amount of DNA, Neanderthal DNA, is the amount of Neanderthal DNA correlated with the difference in endocrine level of different populations?
Endocrine? Is it endocrine or
No, the questions read like this. Is the amount of Neanderthal DNA correlated with differences in endocrine level of different populations?
Referring to?
Hormone levels.
Hormone. So hormone very difficult to estimate from the DNA, but of course this varies significantly, right? Even in a day in the same individual varies. So that all depending upon various factors. So if something very drastically different, that it may be because of the DNA, but nobody looked at specifically how the endocrine of both the human and the Neanderthals. So very difficult to say with the existing Neanderthal DNA, unless otherwise you estimate the endocrine level in the serum or plasma, whatever it is.
So the next question is, how is the study of Neanderthals related to current times?
Related to current?
Current times.
Current times.
Current times.
COVID-19. You well explained that one in the lecture, right? Next question is it possible that we will preserve the DNA of all species on the moon like lunar arc? I don't know. What is... I'm not able to understand the question.
This is a technical question. What are the challenges in the steps of receiving the raw data after sequencing? Is there any problems related to annotations? It's a technical question. Want me to read it again?
Can you read it again?
Yeah. What are the challenges in the steps after receiving the raw data, sequencing data? Is there any problem related to annotation with respect to the Neanderthal DNA?
No, that was initially that not much of algorithm when it was started, 2006, if you imagine. They might have faced a lot of challenges in writing new algorithm to compare with human DNA. So now it is very simple. No, everybody now is different algorithm. Every lab they are using large number of the software which is available online, but there are paid software like Trajan, which can analyze and of course call the variants within 20 minutes of the whole genome. So the technology and also the softwares have been developed such that it's no more difficult to analyze and annotate the genome.
One more question is that last question got corrected now.
Is it possible that we will, that is the one, sir? We transfer the DNA to moon. Is it possible that we will preserve the DNA of all species on moon? Like, I don't know, like they are asking whether we can actually transfer the DNA to the moon. I don't know what they actually mean.
You can preserve the DNA, but where you want to send up to us.
And incidentally, the surface temperature of moon is 60 degrees centigrade, right? No, is it possible
but
to...
DNA is more stable compared to any other molecule, so it can withstand. 60 degrees, okay.
Any other question?
It's okay. So it was a very nice lecture and we enjoyed it. So most of these findings are being always carried out with our individual speculations, and it has helped us to come all the way through. But now with that same speculating aspect, is it a way to predict the future development, at least with the help of these reference genomes, and what are the phenotypic characters which has been evolved and how it is going to be there in the future? That is the one simple thing.
It's very, very difficult to predict the future, right? So what happened in the past, okay, we can see that and presume how it would have happened, right? But we cannot predict the future for many reasons, because the evolution direction can be anything, right? And also, if you predict something that this is what is going to do, and also it is linked with the reproductive nature of that new species, right? Even if you see somebody is very unique, like very tall, right, with a very heavy build, and you feel that, okay, so next generation or the next future is going to be like that. But if there's only one individual because of some mutation happen like that, but whether somebody is ready to marry the person, and once it happens, only that going to be next generation, and the population level is going to increase. Otherwise, it's a kind of left out, and naturally it will be eliminated from the population. Unless otherwise it has the advantage, it's not going to go to the next generation.
Yes, sir. Because why I asked this is we remember studying in early days that usage of organs or bones, for example, the jaw bones. When compared to the earlier Neanderthals, they were using raw meat or vegetables, and then the usage of the jaw bones was much, much more. Whereas in the future, and then now, and then when we imagine for the next another 100 years, the pills are going to give you the energy, so the usage of jaw bones are going to be very less. So they already predicted, and then a few of them have said the jaws will be completely lost. So this was one of the things which we read in our books earlier in future evolution. So like based on scientific evidence,
it's not like that. See, the jawbone and the muscle, it's nothing to do with this kind of activity. In fact, in the early species, they might have used very, very hard material to bite, right? But they have not spoken. So their muscles are much tighter compared to the human, because so that relaxing of that muscle, again due to mutation the genes, have helped the human to speak, right? So as long as we speak, use the jaw muscle, I don't think this is going to happen. And there is also prediction that the Y chromosome is going to disappear. No, that also I don't believe that is going to happen soon. Anyway. Of course, the Y chromosome has reduced drastically, removing all the non-coding sequences, but still has a lot of non-coding sequences. It was predicted that it has evolved from initially autosomes, from the X chromosomes. Now, that's why you see that some of the genes, like the one which I showed, amylogenin, is present on the X chromosome and also the Y chromosome. Same way, many genes are present on both the chromosomes. But most part of the Y chromosomes are having so-called junk DNA, or repetitive sequences. So, but still it has to be there to carry the SRY, sex determining age, on the Y chromosome. Unless otherwise that is present, then there's not going to be a male.
Yes, sir. And then we in immunology we also see that the part of the Y chromosome has been transformed to the X chromosome, and then Y linked a lot of autoimmune disorders has been explained. So if Y chromosome gets disintegrated and go into X chromosome component, then what happens to the integrity of the Y chromosome in future?
No, in fact, the Y evolved from the X. That's what I'm saying.
Okay.
One of the X chromosome become the Y.
Okay, sir. Thank you very much, sir. Thank you.
So we have one more question. As per the Neanderthal Nobel laureate, in modern humans, 1 to 2% of the Neanderthal DNA, which contains serine, is responsible for oxidative stress-related diseases. Is it possible to silence those genes as a treatment strategy for human diseases?
Yeah, there are approaches for correcting some of the genes. Several aspects. One is the gene therapy earlier, stem cell therapy, now using CRISPR-Cas technology to silence some of the genes. That is happening, but the problem is that if there's an off target, if you are targeting one particular gene to correct that, but it might target something else which may create some more problem than curing the problem, right? So it has advantage, but people are not ready to use any of those modern techniques for correcting the genes. But one particular thing was successful in case of mitochondrial diseases. The mother has a mitochondrial disease, Melas or some other disease, and she has the mutation. She doesn't want to pass that mutation to the next generation, because unlike nuclear genome where you have only 50% is going to go, but here all the mitochondria has to go from the mother. So there was one successfully they have done using the donor's egg. What they did, they have obtained donor's egg, removed the nucleus from there, that means the cytoplasm including the mitochondria of the donor, then the mother's nucleus have been introduced there. So that means the egg consisting of only the mother's nucleus, remaining all from the donor and the healthy mitochondrial DNA, and that was fertilized with the father's sperm. So the child was born maybe 5-6 years back, but there's no follow-up.
The question followed by that one is whether that could lead to the increment of average lifetime of humans.
What is it lead to?
Average lifetime of humans.
Average lifetime, but I don't think so, because now we are having much better lifespan. Okay, we don't know the average, but overall it's a better life. That earlier people with 60 years and look very, very old, but now we are seeing somebody is retiring, and you see that so they are not look like 60 years, right? So that's a stage. So that means that average lifespan has been increasing, of course, due to various reasons, but we'll not discuss about that. But we don't know what would be the average lifespan of that particular individual. We'll have to wait and see.
Hello, sir. Good evening, sir.
Good.
Hello, sir. Good evening, sir. It was a nice presentation, and I got too much of information from your presentation, sir. So, and my question is, in your previous initial slide, a person got the morphological features like chimpanzee in the previous slide, sir. Is it related to the atavism, sir? So the gene activation and will get the characters which is found in the previous evolutionary times, sir?
Yeah, so that's what I mentioned, that some of the characteristic features which we obtain due to mutation. So I can tell you some simple example. If you look at our amylase gene copy number, it is multiple copies. But if you look at the early species, one example I can say, the sample which you have analyzed from Europe, okay, 8,000-year-old sample. The amylase copy number there is lesser compared to the modern human. The modern in the sense those who are living today. So because after agriculture came into existence, started rich amylase rich food, starch-rich food, the copy number increased to digest the starch efficiently. So because of there's a need, there's a mutation which gives added advantage, right? So, sorry, I forgot your question.
Yes, sir. Is it related to the atavism to get the...
So, yeah, correct. So, so in this case, what happened that the features which we have lost due to the mutation, if there's a back mutation, and that has to actually get back to the earlier features, right? Like in the experiment we correct, for example, there's a disease, you wanted to see the impact of the disease in the animal model. So we introduce that mutation or knock out the gene and see the impact. Then you correct that and see, regain that the original phenotype. Okay, the same way here there is naturally the mutation happened, due to which we are having a different kind of phenotype, the way we look like today. And if there is a back mutation, I think there could be some master regulatory gene, that mutation happened, that there is a back mutation, obviously the individual have to have the earlier phenotype.
Thank you, sir. Thank you very much.
Hi, sir. Hi, sir. Good evening.
Sí.
My question is, like, we have seen like advantage of having a Neanderthal DNA is like we have better immunity or something like that. So, do we have any disadvantage of it? So, like having a Neanderthal DNA, like we have seen like the French and the Han and the Australian people have DNA. Do they have any disadvantage of it?
No, there is no advantage here, because everybody inherited the genome from our ancestor, like hominids, right? So here we are looking at exclusively how much of the Neanderthal genome that tells that the interbreed between the modern human and the Neanderthal. Because that found outside Africa, there's a mating between human and the Neanderthal. So that's not going to give any added advantage. This is just to show that there was a interbreed between human and Neanderthals.
So, like, one more question is like, why you saying about the tribes? So, you something about the tribes. So, I got like both sentinels, right? They are cut off the world. I mean, they are not. We are not allowed to like interact with them. Has any research been conducted on sentinels like prior, before they were like they banned to go there, or like any similarities between the Neanderthals or something like that?
Now, there are four tribes. One is the Onge, Great Andamanese, Jarvas, and Sentinelese. So we could collect sample from Onge, Jarva, and Great Andamanese, but not from Sentinelese. They are still in isolation. Nobody can go near them. Recently, about 4 or 5 years back, they have killed one, I think, American tourist who was trying to go near them, and they shot with the arrow and killed. So that's the status.
So we don't know nothing about them, right?
We don't know nothing, but since we know something about the other 3 tribes, and we predict that because they are part of the early human migration. Of course, they must have split and settled in different islands. But by looking at the mitochondrial DNA of Onge, Great Andamanese, and Jarva, they all have a common ancestor. So that means Sentinel also should be having that.
That's all. Thank you, sir.
Is there any question? Can we close the session today? Yeah, now we are coming to a close. So, first of all, let me thank Dr. Thangaraj for delivering a wonderful lecture. It was really a good lecture and all people enjoy the lecture. So, Tamil Nadu Science Forum will continue to do such lecture in future, and we expect you to deliver more such lectures for PSL. We will be approaching you again and again. Thank you very much, sir. And my next thanks goes to Dr. Arvindhan, who is leading the Department of Genetics in Madras University. He is very much helpful in organizing the lectures, and he has shared the lectures to his students, and some of his students has come here as well. Thank you very much, sir. So my next thanks go to the library people, and really they provide us a space, and they provide all the amenities for them. My special thanks goes to Mr. Karthikeyan, Sandana Karthikeyan, and the people who are helping us to deliver the lecture through their technical help. Our people who are in the audio system, and they also relay the lecture through the Anna Centennial Library YouTube channel. They are specifically taking a video shot and they are relaying it, and thank you very much for them. And my last thanks goes to all the audience. Without them, we can't have such lectures. They are continuously patronizing us. Thank you very much, all of you. Thank you.
Okay, so thank you from my side to the organizers for having this kind of lectures for the public interest. Okay, so continue to do so. Thank you very much. Thanks to Professor Kumarasamy for inviting me. Okay, so I leave.